We are built out of proteins. DNA is the instructions for making these proteins. But how exactly does that work? How does a string of A's and T's and C's and G's code for blue eyes or the ability to digest lactose? This unit, we will be focusing on gene expression, the process by which a gene determines phenotype. How do organisms regulate proteins? They need a system to start, stop, speed up, or slow down production based on environmental cues. Lastly, different organisms go about gene expression and regulation in slightly different ways, and I want you to be aware of all these key differences. How do we make proteins? There are two different stages. First of all, the DNA that is composed of genes has to go through a process of transcription where it creates an intermediate called RNA, and then that RNA gets translated through a second step into a polypeptide, which is a string of amino acids. That string of amino acids gets folded and turns into a protein, and that protein is what is being expressed as a phenotype. It is worth mentioning at this point some key differences between DNA and RNA. You should be already familiar with these. There are two different types of nucleic acids. RNA is single-stranded. It uses a different sugar, ribose, as opposed to deoxyribose. It has uracil as a base instead of thymine. And lastly, in terms of types of RNA and types of DNA, DNA, we can think of nuclear DNA as most of the DNA in your body, but you also have mitochondrial DNA, mtDNA, and if you're talking about a plant, then there is chloroplast DNA. For RNA, there are some types that you may not be as familiar with. There's messenger RNA, transfer RNA, and then ribosomal RNA. We're not going to talk so much about ribosomal RNA. Our RNA molecules are functional building blocks of ribosomes. So let's talk about transcription first. The enzyme that unzips DNA is called RNA polymerase, and it attaches to the promoter in this stage. Most organisms have a region of the promoter called the TATA box, T-A-T-A. It gets its name because it starts with a repeating sequence of TAs. Bacteria have a simpler version called the Pripnow box. The small section of each gene binds to proteins called transcription factors, which recruit or block an enzyme called RNA polymerase. This enzyme is what synthesizes the messenger RNA, mRNA, required to make proteins. Before we start talking about making proteins, let's think about happiness and cocaine. When you feel happy, it is often because a protein called dopamine is being produced by the brain. It is normally produced in small amounts. Certain activities like eating or having sex can cause genes in your brains to produce more. This dopamine goes from the brain to the nerves to the bloodstream and all over the body, where it has different effects on different cells. For example, receptor proteins on cone cells in your eyes react to dopamine by increasing sensitivity to color and bright light, whereas red blood cells produce proteins that cause the blood vessels to dilate, allowing more oxygen through and thus making you feel energetic and awake. Other proteins break down dopamine to make sure your body stays at a normal level most of the time. What happens when a person takes cocaine? Cocaine binds to the transporter protein needed to ferry dopamine across the nervous system. The brain, noticing that the message is not being sent to its destination, makes more dopamine and sends it. But it just builds up in the synaptic cleft until you get a massive dose of dopamine in your body. Your body breaks down the cocaine eventually, and the feeling fades. Continued use of cocaine tells the body something is wrong. You are not supposed to feel this happy all the time. So it reacts by reducing the number of dopamine receptors on the outside of the cells throughout the body. Now, the things that made you happy before no longer give you the same feeling. And the only way to make you feel happy is by a large dose of dopamine through taking drugs. This is how people become addicted. But we are all different. So one person may have a phenotype with more dopamine receptors or a different reaction to those receptors binding to dopamine on particular cells. My point here is that dopamine is a protein regulated by gene expression. We each produce different amounts when we engage in fun activities. Some of us have clinical depression because our brain cells don't produce enough dopamine or our body cells don't react as much. ADHD is linked to a mutation on a dopamine receptor, another protein in the brain, and the reactions of every cell to dopamine is determined by the DNA in that cell. Back to transcription. You just got an A on your test. Your brain receives that information and sends transcription factors to the genes that code for dopamine production. Once transcription factors are at the TATA box, at the right gene, RNA polymerase starts reading the DNA strand. This is called elongation because reading is actually moving down the DNA sequence and attaching free messenger RNA pieces based on that sequence. These mRNA molecules will carry the code from the DNA to the ribosomes, which will manufacture the protein. This strand being read is called the template strand, non-coding strand, minus strand, or sometimes antisense strand. It is always read from the 3' to the 5' direction, which means that the mRNA is synthesized in the opposite direction, 5 to 3. You might be asking, what the hell is a 3' end? Well, the 5 carbons on the sugar, deoxyribose, of DNA are numbered 1' to 5'. I like to visualize nucleotides as turkeys. The phosphate head is at the 5' end, and the feet are at the 3' end. So take a look at the DNA on the right. If the strand to the left, labeled with A and B, is the template strand, will it be read from A to B or from B to A? Remember that the transcription starts at the 3' end. The last stage of transcription is termination, basically cutting off the string of copied RNA and ending its manufacture. This happens when the RNA polymerase comes to the end of the gene called the terminator. We will talk in greater detail about the differences between prokaryotes, bacteria, and eukaryotes, plants, protists, animals, and fungi in other videos, but I want you to compare these two just a little bit here. Transcription occurs in the nucleus of eukaryotes because that is where the DNA is found. But prokaryotes don't have a nucleus, so transcription occurs in the cytoplasm. The next step, translation, takes place within the ribosomes, which are found in the cytoplasm of all cells. And on that note, we are not done with transcription in eukaryotes. Their genes have sections called exons and introns. The exons code for different parts of the proteins, and the introns, called junk DNA, don't code for anything, which begs the question, what is the point? Why do we have introns at all? Imagine that exon 1 codes for spaghetti, exon 2 codes for meatballs, exon 3 codes for tomato sauce, and exon 4 codes for Parmesan cheese. Because sometimes maybe you want no meatballs and sometimes you want no cheese. Making these ingredients in different bowls, you can mix and match. This alternative splicing means that genes can code for many similar proteins instead of just one. The introns ensure that the ingredients don't get mixed up before assembly. There's some vocabulary involved in gene splicing that is worth mentioning. The spliceosome is a large protein, think about photosystem 2, that orchestrates splicing. The spliceosome is our pasta chef. Of course, a good chef needs a master chef like Gordon Ramsay to yell at them so that they keep working. These are the ribosomes. A sequence of adenosines is added to the end of the spliced mRNA molecule. This is called the poly-A tail, and it stabilizes the molecule. Lastly, a GTP cap is added so that the ribosome has a molecule to grab to start the process of translation. As I mentioned, translation, the reading of the mRNA to manufacture the protein, takes place in the ribosome. It consists of two subunits of mRNA, which runs between them as the gene is read. Reading from the 5' end, where the GTP cap is, transfer RNA molecules that look like twisted paper clips match these anticodons with 3 nucleotide segment codons of the mRNA. Each 3 nucleotides for 1 amino acid, and the opposite side of the tRNA attaches to correct amino acid and string it to the one behind it. The tRNA starts reading at an AUG, which codes for methionine, and ends when it reaches one of 3 stop codons, which cut the strand without adding any amino acids. So all polypeptides start with a methionine, but can end in any of the 20 amino acids. Here is a more detailed look at the tRNA molecule and a table for translating the 3 nucleotide codons into 20 amino acids, plus the stop codons. For example, if a gene reads UUU, that means it is asking for an amino acid called phenylalanine, abbreviated PHE. If a mutation changes the last U to a C, nothing changes. It's still going to produce phenylalanine. There is therefore redundancy in the code, with several different sequences coding for the same amino acid. A change of the last U to a G would code for leucine. If leucine has different properties than phenylalanine, then the protein will fold differently, and like a key that is cut differently may not be able to open the same lock. So mutations can cause the same protein to be created, similar proteins, or completely different proteins, which could be lethal, slightly bad, neutral, or even in some cases beneficial. So let's see how well you understand this process. Using all of the following vocabulary words, describe the process of protein synthesis.